Hey everyone, we are going to go over your first two lab kit labs that you should have already received your lab kit materials for. You definitely want to make sure that you go through those kits and make sure you have everything on the kit materials list. And if you're missing anything, you'll want to contact Carolina Biological to replace those materials as soon as possible. So lab kit labs are different than the virtual labs. The lab kit labs, you are actually performing experiments. Most cases, you're going to be performing multiple experiments a week. You will not want to save these until like the day before because they can be very time intensive. So the first two labs that you have is this one about pH and buffers, and the second one is about macromolecules and enzymes. So we're going to look at the lab manual for the pH first. So it's really important that you understand what pH is, what buffers are. There are going to be some videos in the class announcements, but also your e-text kind of goes into that as well. So this kind of gives you like a little bit of an idea of like the time requirement that's going to occur. And then it gives you like some ideas about when you can do the activities, but that activity 1 should definitely come before activity 2. So the background information that's listed here talks about pH, about looking at, hydrogen ions versus hydroxide ions, what the scale is, what's considered an acid, what is considered a base. You can also determine the pH of a solution using this formula that goes over what buffers are and the equation that's used for preparing a buffer solution and what the pH would be. And then it goes into the materials that you need. And this dilution equation is really important. I'm going to show you why in just a second. So these are the materials that you need from the kit. And then there's always a needed but not supplied. So these are going to be what you have to supply. So you're going to actually have to go out and get distilled water. Not too much. I mean, it's 100 milliliters. It's not too bad. So when we are looking at completing these activities, there's a lot of prep work that goes into doing activity 1 and activity 2. So essentially you are going to have a tray. It's got 4 rows by 5. Make sure that you label them correctly. So the rows should be A, B, C, and D, and then the columns should be 1, 2, 3, 4, and 5. You're going to follow these instructions, but you have to actually perform a dilution. And in order to do that, you have to be able to calculate the volume of sodium hydroxide that you need to prepare a 0.10 molar solution. So it says refer to the dilutions section. That is up here. So in order to calculate a dilution, you have to take the initial molar amount of a solution and multiply it by the initial volume, and then find the final molar amount and the final molar volume. So that looks really complicated. They give you an example right here. So they said, suppose a 1 molar solution of sodium hydroxide is used to prepare 100 milliliters of a 0.10 molar solution. So the first measurement they give you, that's going to be the initial molarity. You are not sure yet what your initial volume needs to be, right? Because we need to know how much of that solution to use. The next, molarity is 0.10, so that's your final. And then your final volume, because you're preparing 100 milliliters, is 100. So essentially you have 1 molar times the variable that you're not sure of yet is equal to 0.10 times 100, which is 10. So your initial volume is that you would need 10 milliliters of that solution combined with 90 milliliters of water to give you that amount. So looking at the volume needed, because you have to calculate the volume correctly of the sodium hydroxide and the hydrochloric acid. So you have 1 molar, you've got 12 milliliters of a 0.10 solution. So essentially you've got, you're basically what you just did, right? So we did 1 molar, you need a 0.10 molar, or you have a 0.10 molar solution, but you're preparing 12 milliliters. So instead of 0.10 times 100, it's going to be 0.10 times 12. So you're going to do that math and you'll find a milliliter amount. And then you just want to subtract it from the information that you have, right? So you'll do the same thing with hydrochloric acid, right? So the 1 molar solution, you're basically going to be doing, that 0.10 times 12, and that should give you your answer. Activity 2 is going to be using test tubes. And you are basically just looking at buffer solutions in each one. There's going to be an indicator and colors that go into the wells. This is just kind of like a simple match up what should be in there, and see, fill out the data table and figure out the concentration of the buffers. Honestly, I would not do I know at the beginning it said you can do activity 2 on a different day. Honestly, I wouldn't. I would just go ahead and do activity 1 and activity 2 on the same day. It makes things a lot easier. And it shouldn't take that long. Like this particular entire lab should take about 30 minutes to complete. And then of course you're going into, after you complete all that, you are filling out the information in your lab report. So you've got you're going to write down what the volume is that you calculated, like I showed you. Then you have your data table. You're talking about what happened. And then for activity 2, your data table, and then your photos. So pretty cut and dried. So then you're going to move on to your next lab, which is about macromolecules. Now this one is going to be a little bit more intensive because there's more materials that you have to gather. So going into your lab manual, macromolecules you should have already learned about from your readings, which is, carbohydrates, lipids, proteins, and nucleic acids. So this is giving you just a quick overview of it. You do have 5 activities in this one. One of them takes about an hour, so you're going to have to kind of be mindful of your time. It does talk about the macromolecules and their monomers, which you do need to know for this lab as well as for your quizzes and exams. When you read through this, make sure you read through it carefully, that you fully understand it. These are the materials that you need from your lab kit. However, you'll see that you actually need a lot of things supplied yourself. So these are the things you have to go out and get. So you need saltine crackers, you need distilled water, you need a container of, tap water. You need carbohydrate sources, so sugar, fruit, any kind of vegetable juice or bread. You need a protein source, so any kind of meat or milk or beans or tofu. I will say that liquids can be sometimes easier to read than foods, but not always. It just depends. you need some food samples. So something different other than that protein and carbohydrate source. Um, you need vinegar, mustard or ketchup, salad dressing, and then 3 foods of unknown fat content. So what you choose is up to you, but you can see, right? Like you do need to go basically grocery shopping for this lab. Um, so it kind of goes through your prep that you need to do. You do need a brown paper bag. Um, and then, you're going to basically go through and test, these different foods for the macromolecule content. So you're going to see how much starch, is in this potato starch solution, but then, you're going to also test, some of the items that you bought, right? And so, same thing with the protein test. So, you don't have to have new foods for every test. So, let's say like you have bread, like you don't need to have different types of bread. Like you can use the same type of bread for the starch test, for the protein test. Same thing with like meat, right? It's like if you have like a slice of turkey, like you can perform the starch test on pieces of the same turkey. You just want to, right? Like you just want to break up the turkey. So you don't want to like do a protein test on top of where you did the starch test. Like you want a new piece of turkey. Um, so, and then the same thing like with the fats. This one though, you do have like where you have some specifics that you have to do, versus like your unknowns that you've selected. And then activity 4 is going to be looking at, digestion of a protein. And that is the one that is going to kind of take some time. So make sure that you look over these procedures really well, and understand, what should be happening and what you can expect. Um, and then activity 5, you're looking at, digesting like how long it takes to digest starch by putting the cracker and seeing how long it takes to dissolve, in your mouth and with water. So, all of these have data tables associated with them. You want to be very detailed in your observations of what's happening, for each one of these, tests. And again, this is going to be in your lab report. So if we look at that, right? So you want to be detailed. Like, is there a color change? Does the consistency change? Is it positive for for starch? Is it negative? Same thing with the fats. Like, is it positive for that? Is it negative? Um, all the tests you want to indicate whether or not it contains that macromolecule or not. Um, so not just color changes, but yes, it has, protein, or no, it doesn't, depending on what test you did. So make sure that you've got like all your photos in there. Um, there's definitely going to be a lot of them in this particular lab since there's so many activities. Um, but if you have any questions, just let me know, and I look forward to seeing your results.